Okay, so continuing our lecture, when we are talking about animals, and animals are, we know, higher eukaryotes. They're not only eukaryote, but they are higher eukaryotes. And when we are cloning, doing genetic engineering in animals, as we discussed in the genetic engineering chapter, we have to go for cDNA rather than the genomic DNA, because in eukaryotes, the genes are first, after transcribed, they have exons and introns, and introns have to be spliced through the process of splicing, and introns will be removed, and then the exonic parts, when they are joined together to make a mature DNA, to make a mature messenger RNA, only then we can transfer them, we can reverse transcribe into DNA, and that will be the exact sequence which we need for the proteins. So this is the general letter sequence. This is the sequence that is generally adopted for the development of transgenic animals, irrespective of any species. The first one is identification and construction of the foreign gene, and then promoter sequences. This is very important to have a proper promoter, because without a proper promoter, if you place a faulty promoter and your gene is not expressed, then there's of no use. The introduction of DNA directly into the pronucleus of a single fertilized egg by various methods. There are a number of, this is the essence of this method. A number of different approaches are used for this purpose. And implantation of these engineered cells into surrogate mothers. Surrogate mothers means the female part has to be hormonally prepared for this, for taking the embryo and for starting the pregnancy, because it's not going to be a normal, original, or you say conventional mating process here. That's why we need it. And then bringing the developing embryo to term. I mean, to find out if the DNA has been stably and heritability incorporated into the genome of the newborn. And then the last part is really important, because you have to make sure that the gene is regulated well enough, because its function in its new environment. Why do we call it a new environment? Because this gene is naturally not present in that particular genome where we are inserting it. For example, if we are inserting a gene of insulin in the mammary glands of a cow, so that insulin is synthesized in the milk and then we can harvest it from the milk. That's not where the environment of insulin gene is. Insulin is going to be in the normal conventional location is pancreas. So that's why we call it new environment. It's very important that it is able to express and it is able to regulate and it is able to propagate it. Okay, the foreign DNA can be inserted using many approaches, but in this lab, this course, and in this lecture, we are going to concentrate more on 3 different approaches. The transfer through lentiviruses, using a virus as a vector. We discussed already in genetic engineering course that this can be done. The second one is microinjection. Again, we also discussed this, that this can be done using microinjection. And then the pluripotent stem cells. We discussed a little about pluripotent when we were discussing in the plant biotechnology lecture about totipotent. I told you we will discuss in detail. In the next lecture, we will specifically talk about stem cells, and there we will discuss pluripotent or pluripotency in great details. These 3 are really important, and the 4th one is also one of the approaches of making transgenic animals, but that's a somatic cloning, so that's a little different. This is the process through which Dolly, the famous sheep, was cloned in 1997 in England. Okay, so okay, now the first part is retroviruses. Retroviruses and lentiviruses, they are the same, they are just different families. They are all inside. Retroviruses are in lentiviruses. They are actually RNA containing viruses and we use them as a gene delivery agent. This is a typical structure of a retrovirus. You could see there are glycoproteins. These are the proteins through which they enter the cells. There is an envelope that is surrounding and enclosing the genetic material, which is actually viral RNA molecule. And inside this package of viral RNA, the genetic material of this virus, there are also a few assemblies of the enzyme reverse transcriptase. And as we know about the life cycle of a retrovirus, it attaches to the cell membrane through this glycoprotein anchors, and then it dissociates, and this part is inserted into the cytoplasm, where it uncoats. After uncoating, the viral RNA is released in the cytoplasm, and through the enzyme reverse transcriptase, this RNA molecule is going to be changed into a DNA, a single-stranded DNA, which will be eventually turned into a double-stranded DNA. And with an integrase enzyme, it's always there, with integrase enzyme, this will integrate into the genome of the invading organism. Now, this is very important that this retrovirus integrates into the genome. If it does not integrate into a genome, we don't get our results. Okay, now, okay, so this is a typical lentiviral transfer vector. As you can see, this is introduced into the packaging cell line, so that it produces the viral proteins, which are very important for the production as well as for the propagation and the application of the transgene sequencing and packaging it into the RNA molecules. And we could see that the transgene is inserted here. Along with the transgene, a promoter is very important, because as we discussed, if the promoter is not functional and operational, this gene would not be expressed. So we need a really important, it's very important to have a proper promoter. And in this vector construct, you could also see a 3' LTR and a 5' LTR. Now the 3 and the 5' LTR are, sorry for that, they are required for the production of the lentiviral RNA in the packaging signal. These long terminal repeats, they are required for lentiviral RNA. What does this mean? This means that when this gene integrates into the packaging cell line, we have to first integrate into packaging cell line. The RNA, this RNA has to be produced, and inside this RNA, we will have a sequence of our, like this RNA. So our gene of interest and the promoter would be converted into a messenger, not in a messenger, but into an RNA molecule, and this 5 and 3 LTR play a vital role in this. This is a phi or psi. Oh, this phi or psi signal is required for packaging the RNA into the viral particles, and these other regulatory elements like Woodchuck post-transcription regulatory element, it enhances the transduction of the host cell and increases transgene expression in animal cells. This is a general structure of the retroviral construct. This is the DNA structure. When this is integrated into a packaging cell line, then the viral particles will be produced, and our transgene would be in the form of an RNA in a packaged retrovirus. Okay, whereas gene transfer methods are used, the use of retroviral, it has some advantage, because it is an effective way of integrating the transgene genome of a recipient cell. Retroviruses, they have RNA genomes which are used as templates for reverse transcriptase to synthesize a DNA copy that can be inserted into the genome. As I said, the genetic material of these viruses are in the form of RNA, which can be converted by a reverse transcriptase enzyme to a DNA. But there is a general limitation with these vectors. because only a small amount or a small sizes of DNA, roughly around 8 kb, can be transferred. Because of a very small size, there is another problem that for eukaryotic genes, you need not only the transgene, but also the regulatory sequences. And for example, this is a regulatory sequence, this is a regulatory sequence. And when you are working with such a small payload, it already produces problems because not every gene can be packed within 8 kilo base pairs. Okay, there is a further disadvantage, but first we have to discuss how does that happen. Now, we have a female donor. Okay, how does it start? We have a female donor. Now, where does the female donor come from? So it's the two animals are mated, and we have a female donor. We take the female donor, we harvest the embryos at the 8th cell stage. It's very important because after this method might not work. So the 8 cell embryo is harvested. We have a transgene already, a retrovirus is already prepared. We already prepared it to using this sequence. So it's like a virus like this, but it's having our sequence, this sequence in the form of RNA, genetic material. And this is already, we have prepared it and we infect it with this 8 cell embryo is infected with the retrovirus containing the transgene, and then the embryo is actually implanted into the female, and we wait for the progeny to be produced. When the progeny is produced, we will find out after the pups, these young pups are formed. We will have to check which one is the transgenic founder. In this figure, you could see this one, the one that with filled circle, red circle, is the transgenic founder. But how do we know? Because when we inject this transgene into the 8 cell embryo through a retroviral vector, we will have to implant this embryo into the female. And we do not know what happens next, only when we have pups. And when the pups are formed, even then, from the look, we can, well, sometimes we can find out, but there are molecular and there are breeding methods which are very important to do. I will discuss in the end. As I discussed earlier, there's a higher chance of toxicity as an advantage. These viral vectors, though they are defective these are defective retroviruses, but they can always reactivate and they can cause a lot of toxicity. There's a very strong immunogenic response against these viruses because these are viruses at the end of the day, and the low loading capacity. The next method is microinjection method. As we discussed earlier, we are going to have or emphasize on 3 parts. Okay, because of the disadvantage of the retroviral method, where we could have toxicity issues, we already have a very big issue of the size, only 8 kb. So if the gene is big enough, is small enough, only then we can transfer it, as well as we need to have the regulatory sequences, so 8 kb is not enough. And also the immunogenicity is a problem. So the number of this procedure is going to be operated in such a way that you have a super ovulated female. Now, how we super ovulate female? The female organisms, they are given injections, initial injection of pregnant mare serum, and then after another injection 48 hours later, human chorionic gonadotropin. These are 2 hormones. So these 2 injections in the due passage of time will make the donor female to super ovulate. It can produce about 30 to 35 eggs, which is the result of the super ovulation, because a normal a normal mouse would produce only 5 to 10. So you could see there will be an increase 7 to 10 times, 7 to 8 times, 5 to 40, almost 35, 7 times, yeah. Then these super ovulated females, they are mated so that the eggs, they have so many eggs, like 35 eggs, they become fertilized. They are, then the next step is a little harsh, because then we kill these female super ovulated and mated females. The fertilized eggs, we rushed out, take them out from the oviducts, and then we do microinjection of these fertilized eggs. Sorry. So now we can see these are super ovulated female. They are mated with normal males, and as after the fertilization process, when the super ovulated females are with having a number of eggs, receive sperms from the male donor, these rats or these mice are killed, harvested. We harvest the embryos, and then what happens, there is going to be in this fertilized egg a female pronucleus and a male pronucleus. You know that the female pronucleus, it does not contain any DNA. The major part of the DNA, well, it contains DNA, but major part of the DNA, the haploid DNA, is coming from the male. What happens now? We have a suction holding pipette. So to one end of the embryo, we will have a suction pump, and to the other, we will have we will inject through a small pipette. All this will be under a camera. You could see this with your eyes if your micropipette is going inside the male pronucleus or not. And in this pipette, we will have a transgene, and the transgene will be pipetted directly into the male pronucleus. Once the transgene has been pipetted inside, injected into this male pronucleus, we will implant this in a surrogate mother. So now, foster mother or surrogate mother, these will be again female animals. They would be initiated to start their pregnancy cycle through hormones. So it's a false way to do it, because we want them to nourish and give rise to the transgenic pups. And then after this, we will have the pups, and one of the pups will be carrying, or more than one of the pups will be carrying a transgene, and we will call it a transgene founder. But in this case, again, we have implanted female, and then there are 3 pups. Which one is the transgenic founder? Again, we mention it here is this guy, but how do we know? We do not know. There's a certain process for this that I will discuss in the next few slides. Disadvantages of microinjection. There are obvious disadvantages of microinjection method. We could inject the DNA, and it could integrate at random sites of the genome. Or we could have multiple copies of the injected DNA incorporated at more than one site. This is something we do not want. We want it to integrate at a place, at a site where the regulatory sequences could make the transgene express. Again, same problem. You will not find them in all of the pups and all of the young ones, because in some cases the transgene will not be expressed because of why? What's the reason? It could be integrated in a wrong place. So it's all about chance. And then the copy number may be excessive, and that may lead to overexpression. So you do not want overexpression. If you have a number of, because when we pipette it into the male pronucleus, we just do not have only one transgene. We have numbers of transgene in the solution form and inject. So if more or a number of transgene gets integrated, there might be an overexpression, and this could lead to the disruption of the normal physiology of the animal, and this is not good. The fourth, I should put a number 4 here, the 4 disadvantages disadvantage of the microinjection is efficiency. For example, if you could see, we have a comparison here between cattle, pigs, sheep, and mice. All of them are injected. Now, after implantation, we could see within cattle, it's just about 10%. Within pigs, it's around 60%. Above 80 is sheep and mice. They are roughly 68%. But then when the pups are born, you could see a drastic drop. Now from, I would say 10, we jump to 1 or 2 in case of cattle. In case of pigs, we jump from 60 to 8 or 9. And the higher is coming in sheep, the higher percentage of the newborns with the transgene. Yet it's very low in comparison to the implanted ones. Similarly, mice, and you could see now they are winning the race here around 12% or 16%. But even then, when the live one is born, we do not know if the transgene is going to be propagated. So the transgenic progeny, if you could see, 1% in cattle, roughly 1.5 to 1.6% in pigs, the same ratio in sheep, while mouse or mice is somewhere at 3 to 4%, which means this process is not more efficient than 5%, which means if we have 100 experiments and we try to produce 100 transgenic animals, only 5 times is going to be successful. Now that's really, really low, this efficiency. And this is the problem with production of transgenic animals.